Hello everyone, I am Shengguo Zhao from the Chinese Academy of Agricultural Sciences. It is a great pleasure to share with you our newly published paper in iMeta, titled, Leveraging Core Enzyme Structures for Microbiota Targeted Functional Regulation. Urease is an example. As we all know, microbiomes play a crucial role in environmental ecology, human and animal health, making microbiome research a cutting-edge field of global interest. Although extensive studies have been conducted on the taxonomic and functional diversity of microbial communities, effective methods for precisely modulating target microbial functions remain limited, often suffering from poor specificity and off-target effects. In livestock farming, for example, microbial Urease in the rumen is essential for urea metabolism. However, excessive Urease activity leads to low nitrogen utilization efficiency, necessitating selective inhibition of Urease activity. To address this, we developed a structure-driven targeted functional modulation approach for microbiomes. This method involves identify dominant enzyme genes and their genomes based on high abundance, sample prevalence, and enzymatic activity, resolve the protein structures of these dominant enzymes, perform structure-based virtual screening to identify high-efficacy modulators, and validate their functionality through in vitro and in vivo simulation systems. To validate this framework, we focused on inhibiting rumen microbial Urease activity. We collected 88 rumen fluid samples from cows fed 10 different diets and sequenced the EC gene. Our results showed that diet significantly influenced rumen Urease gene diversity, with 44% of Urease genes belonging to unclassified taxa. Based on relative abundance and sample coverage, we identified 9 dominant Urease genes, most of which originated from uncultured microbes. Through metagenomic sequencing and assembly, we obtained metagenome-assembled genomes, MAGs, and identified the MAEG257, harboring the dominant Urease gene. MAEG257 contained complete Urease and urea transporter gene cluster. Functional analysis revealed that MAEG257 utilizes starch to produce propionate and lactate, while coupling energy and carbon skeletons with ammonia released from urea to synthesize microbial protein. Metaproteomic profiling confirmed the high activity of MAEG257 Urease. Phylogenetically, MAEG257 belongs to an unclassified Sacchini-Vibrianaceae lineage and is widely present across ruminant species. Further investigation into subunit interactions uncovered a nickel transfer mechanism essential for Urease activation. Cryo-EM structural analysis of the dominant EC protein identified 5 critical active site residues governing enzymatic activity. Leveraging this structural insight, we performed molecular docking screening of a natural compound library and identified epiberberine, a plant-derived alkaloid with 30-fold stronger inhibitory potency than conventional inhibitors. In vitro rumen simulations confirmed its efficacy in suppressing urea degradation and slowing ammonia release. Comparative binding studies with sulfur-containing compounds and boric acid revealed that epiberberine interacts with both the flap region and nickel binding site of Urease. Its 2-methoxy groups form hydrogen bonds with key residues, sterically hindering urea access and elucidating its inhibitory mechanism. In summary, this study establishes a novel methodology for targeted microbiome modulation, exemplified by the discovery of a dominant rumen Urease and its potent natural inhibitor, epiberberine. Our framework bridges correlation-based research with functional control, offering a blueprint for precise microbiome engineering.